Hi, this is John Keller from the Department of Chemistry and Biochemistry at the University of Alaska Fairbanks. My seminar today is on investigating the mystery methyl of the fungal amino acid AIB. AIB is this amino acid right here, and the methyl group in green is the mystery methyl. It's produced by the fungus Trichoderma viride. This shows a picture of our culture that we used in this study, where we grew up the fungus, isolated the amino acid, and studied its property, trying to find out how this methyl gets onto that carbon. Aminoisobutyrate is derived from isobutyric acid. That's this part here, and the amino group attached to the alpha carbon. It's related to alanine, which has a hydrogen on the alpha carbon. And an organic chemist looking at this would think that the synthesis of this from an organic chemist point of view would be fairly straightforward. However, the biosynthetic route is not known and is not published as far as I know. My look at the literature, in particular the carbon isotope data that I'll tell you about, suggests an alpha-methyl transfer mechanism, which the organic chemist would illustrate with curved arrow notation, pulling off the alpha hydrogen, making a carbon anion, carrying out an SN2 substitution, kicking out the leaving group to add the methyl group to the alpha carbon. So later in the seminar, I'll tell you about how we made the alamethicin peptide from Trichoderma in the lab, isolated the AIB, and studied its isotopic content. Before I get started, I'd like to make several acknowledgments, especially to the UAF Department of Chemistry and Biochemistry, who provides fantastic research facilities here in terms of instrumentation, chemical supplies, and not least the colleagues on the faculty and students. I'd like to highlight Carl Murphy, who is in charge of their beautiful NMR spectroscopy lab, which I used in this project, and we'll show you the results, some of the results from that. And also Tim Howe down at the stable isotope facility, who did isotope determinations. So I'll start out by telling you about where AIB is found in nature, something about carbon isotopes and how they're measured, a computer modeling of a possible enzymatic AIB synthesis, and then in the last 10 slides or so, isolation and purification of the alamethicin and the constituent AIB and its isotopic analysis. Some of this has been presented in a poster at the 2021 ACS meeting, and the poster is in the hallway outside of 143 Reichart. Alamethicin is a 20 amino acid peptide that's produced non-ribosomally. In other words, it's not encoded by a gene. There's an enzyme that puts this together, amino acid by amino acid. 7 out of the 20 residues are AIB. It has been established that the AIB residues do not arise by a post-processing of an alanine peptide. They actually are grabbed by the synthase and inserted into the peptide. What I'm interested in is what is the source of the AIB. Alamethicin seems to form a 5-helix bundle in terms of its biological function, which is toxic for target microorganisms in the soil environment. Makes this helix bundle which distorts and destroys the function of the target of the membranes in the target microorganisms. It's very hydrophobic, very tight helix. You can see all the hydrophobic groups here on the outside of this helical bundle. Another place that AIB occurs is in extraterrestrial meteorites. And the famous Murchison meteorite and other ones, aguas, that have been found in Antarctica, contain substantial amounts of AIB. So this is the HPLC of derivatives of those. and AIB is peak 16. The most common constituent is glycine. L-alanine is there, and L-glutamate also. So Dan Glavin at NASA Labs has done a lot of research, and also his postdoc Elsilla. And we'll see some of the other data that they have on these amino acids as well. And so analyzing these by their carbon isotopes it's a very powerful tool for the planetary scientists, for enzymologists, for chemists around the world. Biologists use isotopic ratios as well. And the most common way that these are reported is as what's called the del C13, and that is a ratio that reports the isotopic ratio as relative to a standard. So in other words, del C13 is the difference between the 13 to 12 ratio for a sample and the 13 to 12 ratio for a standard, all divided by the standard, and that times 1,000. These turn out to be very small numbers, so you multiply by 1,000 to get it into something that looks reasonable, like 20, 30, minus 20, minus 30. I do the math here. You see this in a more common form. If you look in a textbook, it'll be the ratio of the ratios minus 1 times 1,000. Same thing. Carbon isotope ratios are determined in an isotope ratio mass spectrometer. And typically what happens is the sample, the amino acid, is combusted to produce CO2, and that is shot into the source of a mass spectrometer. These IRMSs are tuned specifically to the isotopes of carbon dioxide, and they measure the intensities of several different masses, mass to charge ratios, 44, 45, and 46. And of course, 44 is the all 12 light. 45 has the C13 in it, but it also has some oxygen 17. And so the computer program will take these three intensities, subtract out the effect of the oxygen, and give the true 13 to 12 ratio. On Earth, either biological or geological or meteorite samples, the percent C13 ranges from about 1.04% all the way up to 1.17%. Very, very tight range. But the fact that the mass spectrometer can measure these out to the 5th or 6th decimal point means that we can make hay out of out of these very similar ratios. They are very significant and are characteristic of all kinds of different physical and biological processes. The standard that's used for these is the so-called PDB, which is a limestone from the PD formation, and it's called belemite limestone, so PDB for PD belemite. Has a rather high 13 to 12 ratio compared to biological samples. Carbon isotope ratios from fungal peptides and meteorites. Dan Glavin from the NASA labs and his co-workers were interested in proving that the amino acids in meteorites are of extraterrestrial origin. They determined that the carbon isotope ratios in these extracted amino acids and compared them to amino acids from the soil fungi, and in particular the peptides from those soil organisms. Alamethicin is one, and a couple of others from different species. And of course what they found was that there's a huge difference in these ratios from the meteorite ones versus the biologically derived ones. Clearly the amino acids on meteorites are non-terrestrial in origin. In looking at this table, however, I noticed a interesting dichotomy or difference between the AIB ratios and the other ones, regardless of which organism or which peptide they came from. They're all range from 5 to 8 per mil lighter than the normal amino acids, glycine or alanine. And this got me to thinking, well, why are the del C13 values of fungal AIB consistently low by 5 to 8 per mil? Well, one possibility is that the AIB actually does contain the alanine-derived carbons with the normal ratio, but then include a very light methyl carbon. So for instance, this would entail the alanine with a del C13 minus 18 per mil, reacting with some kind of AIB synthase, attaching a new methyl to it, which would result in this minus 23 per mil. And in order for that to happen, this new methyl would have to be very light, minus 38 per mil. You can see the weighted averages of minus 38 and 3 minus 18 gives you minus 23. This sort of a light methyl would arise from a methyl transfer reaction with a large normal 12 to 13 kinetic isotope effect. And it's well known that the lighter carbon reacts faster in many different reactions. It's called a normal isotope effect. We'll show exactly how this can happen in this case. Turns out this is quite a reasonable value for a new methyl inserted by a methyltransferase. So in thinking about this type of a reaction as an enzymologist, what kind of a nucleophile would one see in an enzyme? And the all-time favorite is a vitamin B6 cofactor attached to various decarboxylases, transaminases, and others, is a pyridoxal 5' phosphate. So this has a main feature of this is a reactive aldehyde group here that reacts with amino acids. Adds on here to form a imine bond after a dehydration reaction. And this, in effect, connects the alpha carbon with this electron withdrawing group in the cofactor. And so, for instance, in this case, if an enzyme would pull off the hydrogen, as we suggested before, to make an anion here. Now, in this, with the attached cofactor, this stabilizes the anion and makes it much easier to remove the hydrogen compared to the amino acid by itself. Now, what kinds of methyl donors might one see in a biological system? The best candidate would be related to 5-methyl tetrahydrofolate. That's this whole thing right here. It's a well-known cofactor involved in 1-carbon transfer reactions. And for this to be an effective leaving group, this nitrogen must be protonated. Another possibility is actually more commonly found as a methyltransferase. It's S-adenosyl methionine. Like I said, this has been known in many different DNA methyls, RNA methylation, et cetera, et cetera. The problem is that these occur without the intervention of the anion-producing functionality of vitamin B6. Based on those ideas, then, I went ahead and constructed computer models to see if this methyl transfer reaction is energetically feasible. And so when you typically do that, the theoretical model must preserve the chemical function but minimize the number of atoms, because when you do a density functional theory or quantum calculation, the number of orbitals that you are required to calculate increases tremendously. And so you have to, even with a model of this size, the fairly low level of theory, density functional theory, was utilized. So for this one here, these alkyl substituents were removed. They're not really important in the electronics. However, the OH was left because it plays a very important role, hydrogen bonding to the nitrogen. Sometimes this hydrogen is actually attached here to make a positive charge with an O minus there. And of course, the hydrogen must be on here to provide the positive charge in the ring system. A guanidinium ion was added to model the effect of arginine side chain. We'll see that in a prototype, in an existing enzyme that plays a major role. And of course, the side chain here on the folate is not particularly important electronically, so just preserve that whole thing with the protonated in-methyl structure. And on the studies, which I won't report on here anymore, the trimethylsulfonium was used as the methyl donor in those enzymatic, those model studies. So it turns out there's an enzyme, serine hydroxymethyltransferase, that incorporates a folate cofactor and vitamin B6 and carries out 1 carbon transfers to the N5, involving N5 group here. And so this has a interesting structure here. I mentioned that this is the imine derivative of glycine, which is the normal substrate for this. It's complexed with an arginine side chain. Is hydrogen bonding to aspartate to stick that proton on that pyridine nitrogen. The crystal structure is of a 5-formyl tetrahydrofolate. There's a double bond there. This is a non-reactive in the SHMT reaction because it's oxidized, oxidation state. But still it binds, you see, and they can form a crystal structure and study it that way. Here's a moving model of this same thing here, just so you get a little better perspective on where those atoms are. You can see one thing you can see here is the effect of the tetrahydrofolate makes the pucker in that ring structure and puts the substituent axial, and then that in turn forces the nitrogen substituent down towards the cofactor. Also, you see that this carbon, which would be, say, a methyl group in a methyl transferase, sits directly above the alpha carbon of the imine. So it's in a perfect location for this. So I use this as a model for a starting point for the model calculations. Of course, the models that I calculated had no protein on there, or they are also missing the side chains. So how does the hydroxymethyltransferase itself work? Well, in the normal reaction, the glycine forms this Schiff's base. One of its hydrogens is pulled off to make an anion, and that reacts with formaldehyde, and nucleophilic addition to the carbonyl of formaldehyde. For the formaldehyde, it arises from the folate cofactor that's here. It's not shown here, but this is this or something like it anyway is in there, very close to this carbon, adds onto it. This is called an aldol reaction to give the serine imine, and that's hydrolyzed away to give L-serine. This is the source of L-serine inside cells. This regenerates the B6 cofactor. Well, all right, so say we had a serine hydroxymethyltransferase-like enzyme in the Trichoderma. How would this work? Well, of course, it would have to react with alanine. In fact, it would have to be a D-alanine, because this requires that the hydrogen that's removed, it's gone off here already, but it has to be up and for it to be pulled off by the base. The base in this enzyme, in these enzymes, is on this upper surface right here. And that then, like I said, would react with this in-methyl donor to give the AIB imine hydrolyzed in the same fashion to give the free AIB. So now this was done at the density functional theory with Gaussian 16, building in this anion, adding the in-protonated methyl compound, moving the carbon closer to the alpha carbon, and using the transition state searching function in Gaussian to find this maximum on the energy curve going from reactants to products. So you can see here, this is the transition state. So this methyl group is now flat. It's in flight from nitrogen to carbon. And the product here now, it's fully transferred, and it has a tetrahedral carbon at this point. That new methyl group is in what's called the pro-S position, and the original is in the pro-R position. So you see, this one then would be the one that we would think would be the new mystery arising from transfer from nitrogen. The pro-S and pro-R nomenclature you may not be aware of, and it applies to molecules where there's two identical groups or atoms bonded to a tetrahedral atom, and then two other identical groups. In a chiral environment, these actually are different from each other. And the way this is done, the way these are distinguished is by assigning a heavier isotope to one of these, such as that one, applying the Kahn-Ingold Prelog RS rules to that. And so if we do that, then this nitrogen as first priority, carbon with two oxygens as second priority, and the heavier carbon here as third priority. And we go 1, 2, 3. That's counterclockwise, or that's S. So we say that the green methyl here is the pro-S methyl. Of course, the other one, pro-R. So when we calculate the relative energies of these, we see that combining the two reactants is uphill from a free energy point of view. This is mainly due to the loss of entropy making a complex. But then the actual methyl transfer to the transition state here is as a 19 kcal per mole barrier to that reaction. And then once you form the product here, this is quite exothermic by 12 or 13 kcals per mole. So the fact that this goes down in energy so far means practically that this, if it goes by this reaction and you do attach a methyl, that methyl is stuck on there. It cannot go back over. So it would be for all intents and purposes, irreversible methylation. This size of a activation barrier is actually reasonable for a non-enzymatic reaction. But for enzyme catalyzed reactions, we would be looking for activation barriers in the 10 to 12, 8, 10, 12 kcals per mole, because these are very, very fast reactions. So in other words, catalysis is required in some way, perhaps squeezing them together, for instance, to lower this barrier. You can also calculate an isotope effect here. This is using the PyQuiver program. And this predicts that there will be, that the C12 methyl will be transferred in this reaction 9.5% faster than the C13. That's a 12 to 13 rate ratio, isotope ratio, called kinetic isotope effect, 1.095. And that therefore this pro-S carbon will be enriched in C12. This assumes that the overall isotope ratios of the reactants is the normal, at the normal value from the reacting amino acids. Taking a closer look at what exactly a 12 to 13 isotope effect is, we look at the energetics of a simpler example here. This is an SN2 reaction between chloride ion and a protonated in methyl compound, similar to the reactant that we've seen. Of course, this would start to form a bond, transfer the methyl to carbon through via the transition state. This is a symbol for a transition state to give the final product. When you're doing this, you know, like in all of these kinds of studies with computational chemistry, essentially what you're doing is finding the lowest energy geometry on the potential energy surface. So when you do an optimization in Gaussian, it slides down this until it reaches the bottom of this curve, and that gives what's called the electronic energy. And this is the lowest energy state. However, okay, and that's what this looks like, or this reaction. A nitrogen carbon bond 1.51 angstroms, the chloride ion poised to react out here. Also the nitrogen carbon stretch frequency here is 953 wave numbers. Turns out that these molecules are vibrating even at absolute zero. And if we take that vibrational energy into account by calculating the 0-point vibrational energy, then the actual energy of the molecule, the minimum energy, isn't this one that's right at the bottom. It's up by the 0-point vibrational energy. This is calculated by Gaussian, by adding together all the vibrational frequencies and multiplying by a constant. This is called E0. Well, what happens when we have C13 there? Well, first of all, the geometry is exactly the same. The shape of this potential energy surface does not change when you add a neutron to one atom. But what does change is the vibrational frequencies of the CN bond. It's because it's a heavier atom, vibrates slower, and the CN bond is 946 wave numbers. And when you add that into this equation here, you get a lower 0-point vibration energy. This is the source of the kinetic isotope effect, because when you look at the transition state, the CN bond is gone. It's replaced by something, a motion that is not a vibration. It's a translation of the methyl from nitrogen to carbon. So it's missing from the 0-point calculation. And as a result, these two isotope energies are very similar. They're not exactly the same, but they're much closer than in the starting materials. So qualitatively, you'd say, oh yeah, I can see why 12 goes faster than 13, because this distance, because these are separated wide here and almost together up there. This is a shorter distance, and therefore K12 is greater than K13. You can quantify this by comparing the 0-point energies. These are, again, calculated in Gaussian, and plugging them into the rate equation that uses the free energy of activation in the exponent here. So this is the rate for the 12 isotope. This is the rate for the 13. We take the ratio of those. Then what we actually get here in the numerator of the exponent is the difference in the 0-point energies. This gives a kinetic isotope effect for this reaction of 7.4%, 1.047 for the ratio of it. Okay, given that the Trichoderma viride contains a gene for a serine hydroxymethyl transferase-like enzyme, and that the methyl transfer step has a kinetic isotope effect of 1.095, how would that translate into a del C13 of -38 for an incoming methyl group? And to answer that, we need to use a kinetic simulation program. The one I use is called Tenua. It's Java-based. And it involves defining rate constants for all the steps in a react. This is a fairly simple enzyme mechanism. The free enzyme combines with the substrate. to form one complex. That forms another complex, which is converted in the isotopic sensitive step to the product enzyme complex, which then spits out the product. And so what this program does is model, predict over time the evolution of this, the consumption of the reactant, the production of the product, and the status of all these intermediates as they go. You can define these reactions in two different ways. The normal laboratory enzyme assay is what I call a closed system, where there's a limited amount of the reactant, and over time, say seconds, then the A is used up and goes to 0, and B is equal amount of that by stoichiometry forms after 600 steps. If you also, this program will tell you about the concentration of the different enzyme forms, and these depend on exactly what rate constant you choose for this reaction. In this case, the main thing you can see here is that these jump up to a initial value, and then as A is used up, they fall back in. These equilibria go back to the left, and you end up with pure free enzyme with no intermediates. Now, this is not the way enzymes work in cells, and this is what I call an open system. In an open system, the enzyme uses an amino acid substrate from an amino acid pool, which is continuously replenished from outside by the nutrients flowing from the culture medium. And these amino acids go on to different processes. Some are excreted in different forms as waste. But that's what this enzyme, this AIB transferase, AIB synthase would be working with, would be a constant source of the reactant A. So here, if we don't worry necessarily about the fact that the AIB would be increasing, if we just kept A constant here, that's A. Same rate constants and everything here, except that now we're saying that this is continually replaced. Well, what's going to happen is that this would crank out AIB forever at a constant rate. That's not what happens because clearly these peptide synthases grab it and put it into the peptide and then excrete that outside the cell. But from a point of view of the enzyme forms, these jump up to a certain value, the same one as there, but stay that way in a steady state throughout the time period that you're looking at. Okay, so that's for a simple reaction. What about kinetic isotope effects? Well, to do that, you just set up another series of reactions with a heavy substrate. And somewhat simplified because all these steps here in black are not isotope sensitive. And all you have to do is put in rate constants here that reflect a certain isotope effect, like we calculated back with the densital functional theory. Well, this is the C12 transfer step. This is the C13 transfer step. And you run the simulation the same way. And of course, this being an open system, what we're interested in, both A and B concentrations are constant. So in other words, the isotopic ratio of the amino acids in the pool is what the enzyme would be dealing with. Again, I'm not going to go into the actual numbers of the rate constants, etc., but we can illustrate this process by an energy scale here, where these minima here represent the different forms of the enzyme, EA1, EA2, EP, and E again. And the tops here represent the transition states going from one form to the other, where this red one, of course, would be the one we're interested in. And it turns out that different enzymes have different heights of the isotope sensitive step relative to the other steps. And that this affects the observed kinetic isotope effects in an enzyme reaction. Often these are less than the intrinsic isotope effect. That number that we calculated before, 1.095, you'd call an intrinsic isotope effect. But it turns out that the observed isotope effect is includes a term here called the forward commitment. Forward commitment is a function of rate constants that it reflects what happens to this EA2 intermediate. If this is a low barrier, then these tend to go forward instead of backwards. And that's called a large forward commitment. And say if this barrier is high, well then this EA2 will tend to go backwards instead of forward. It has a low forward commitment. So when you crank in these numbers here, say that this number was large here, then what happens is that the observed isotope effect basically goes away. You can see why that's true, because if every molecule of the substrate, when it gets to this point, goes forward, regardless of which isotope is on there, then you're going to get a low or no isotope effect. If this forward commitment is very small, then this reaction will be very selective in which ones get over this. The ones with the C12 are much more likely to traverse this compared to C13. In that case, you see almost the full isotope effect. Well, so how would we achieve then a del C13 of minus 38 for the product methyl group? Well, there's different ways this could happen. And for instance, say that the intrinsic isotope effect was 4%, 1.04. If the forward commitment was 1, then that would give it to you. If it had a very low forward commitment, that methyl group would have a minus 50 or minus 55 per mil isotopic content. If you had a more like a 9% one, then the forward commitment is going to have to be out here someplace, 3 or 4, to give the minus 38 per mil. I didn't do those calculations. I perhaps should have. Another thing this shows is that, even if you had a zero forward commitment, well, a small forward commitment, you would not, a 1% intrinsic isotope effect is not going to cut it. You got to have a substantial isotope effect, regardless of the forward commitment, to get a minus 38. But the point is that this is certainly possible with a variety of kinetic schemes for the enzyme. So Asila's study of alamethicin showed that the AIB contains an unusually low 13 to 12 isotope ratio compared to the other amino acids. This ratio could result from a biosynthesis of AIB via reaction with a large normal kinetic isotope effect. And using densital functional theory, we showed a methyl transfer to the vitamin B6 alanine anion is energetically feasible. It's exothermic, and it has a low-ish energy of activation, reasonable energy of activation, and a large intrinsic kinetic isotope effect. Probably a AIB synthase by this route would be homologous to known serine hydroxymethyltransferases. Okay, now what have we done in the lab to approach this? And the overriding fact here is that the isotopic analysis of biosynthetic AIB must begin with an amino acid obtained from fungus grown in an AIB-free medium. The commercial alamethicin may be contaminated with synthetic AIB, chemically synthesized AIB, because it's known that you can increase the yield of alamethicin in Trichoderma and other fungi by dumping extra AIB into the medium. Also, if you ask the companies such as Sigma, If they do that or not, they won't tell you. It's a trade secret on how they make their alamethicin and all their chemicals, as a matter of fact. So the bottom line is you got to make your own, and that's why I've spent a good deal of time doing this and figuring out the easiest way to doing it. Another reason is that it's damned expensive, nearly $1,000 per 25 milligrams. Here's the culture of the forms cool little puffy forms of fungus. This is a 3 or 4 day growth. We filter off the white stuff and keep the supernatant because that's where the alamethicin is. And there's a plate where it's growing. If it's a young growth, it's white. If it's older, it starts to make green spores. This is 1 incubator's worth of, gives about 2 liters of supernatant. After trying several literature methods for purifying this, I developed a new method that's actually much easier and gives NMR pure alamethicin in 1 step. And the way this is done is to filter the supernatant through a C18 silica extraction column. It takes several hours to do this. And then you wash this with deionized water and then elute the bound alamethicin with a water methanol gradient. And this gives, like I say, essentially pure alamethicin. Here, this is a small one. I did buy some alamethicin, and that's the standard. Using our beautiful 600 megahertz NMR, I've obtained a number of NMR spectra of purified alamethicin, and this gives exactly the right carbon spectrum. I'm showing the carbonyl region here. Turns out it should have 23 peaks. It's actually got 22 because 2 of the glutamines end up at the same. So this is two carbons here. And I know that this is the right spectrum because back in the day, Yi et al. Published the C13 spectrum of alamethicin. And I first looked at this and I said, oh my God, we didn't get it. But then I noticed that in fact, these small peaks here line up exactly with the peaks in my spectrum. And each one of these is surrounded by a doublet. And these are created by the coupling between the carbonyl C13 and C13 and the alpha carbon. This peptide was produced by growing it in an all C13 medium. And most of the amino acids in there have multiple C13 constituents. So you can see this. Here, these 3, you see there's sets of doublets. That's the carbonyls of the unlabeled amino acids. Okay, well, it turns out that originally I was hoping I'd be able to jump right in and do an isotopic analysis, probably by NMR, of the Pro-S and the Pro-R carbons. That may be possible, but I haven't gotten to that point yet. So taking a step back, we can say, well, okay, if the Pro-S methyl is minus 38, then together the 2 methyls should be what? Minus 30. Still detectable. And that's an easier experiment. I still haven't done that, but that's actually my next step. Well, okay. How about if we just analyze all the carbons with an isotope ratio mass spectrometer, and hopefully this will at least duplicate what was in the literature determined by mass spec. And my method for doing this on this large scale is take the alamethicin, destroy it by boiling in HCl to its constituent amino acids, separate the AIB mostly by ion exchange chromatography, and then take that and analyze it with the IRMS. The ion exchange process is a classic experiment in chemistry developed decades ago by Stein, Moore, and Herz. Stein and Moore are Nobel laureates for their work with RNase enzyme. But they figured out how to separate all of the amino acids, the physiological amino acids, on an ion exchange column. And this is a truly heroic experiment. The column is 5 feet long, and the elution takes 4.5 days. Well, my column is only 37 centimeters tall, as big as I could get, and it takes 8 hours for it to do this elution. I'm just showing the section here where these are analyzed by NMR, each one separately, where you quantify and identify constituents. But it's mostly separated from the alanine. And the glycine, you see, is separated about the same as on their column up there. That's glycine. Alanine. There's alanine. If they had AIB in there, it would probably come off about in here someplace. Of course, it's not a physiological amino acid, so they didn't work with it. But that's the one that then I wanted to stay away from the alanine, you see. I want to get the AIB out of there. The fact that I have not taken the whole peak here turns out to be crucial. So in all, I've done this shows about 10 isotope determinations at the Stable Isotope Lab. And so here's the Del C13 scale. These were all done in triplicate, and the standard deviation of those is about half the size of these dots. Incredibly precise determinations. So the differences you see here are very significant. And in fact, also the fact that 2 of these are virtually identical is also significant. So for instance, this is DL-alanine out of the commercial bottle. One of these is the actual sample. The other one is converted to the hydrochloride, dried down, and then analyzed. They give the same isotope ratio, which they should. But I did that just to make sure that when we took samples from the ion exchange column, which come off as hydrochlorides, that we're getting a reliable isotope ratio. Now, commercial AIB is down here. It's somewhat lighter. These are then the amino acids obtained from the ion exchange column. So these peaks right here refer to a sample of AIB, commercial AIB, where the peak, different sections of the peak, the early part, the middle part, and the late part, were separately analyzed. And they give huge difference in isotope ratio. There's a fractionation that is happening on chromatography. It's amazing to think that a single extra neutron in one methyl is going to make that much of a difference. Well, that's not true. The carbons may be in different places, but still, this is a large effect considering that these are just heavy nuclei. It obviously has something to do with the vibrational properties of these molecules. Anyway, if you average these out, you'll get that number, and that is almost exactly the same as what you got from the original bottle sample. I did another one where all I recorded was, all I collected was a peak from the middle, and that gave a minus 31 value. So I'm saying, well, okay, there's the bottle value, and if you were to take these fractions and add a correction factor of about, what, 0.8 of a per mil, then you'd get the correct bottle value. So what about this is now the AIB from the alamethicin. I showed the chromatogram of that where I took only some of the fractions. Because of the fractionation that happened, you see, this is not the true isotope ratio. It has to be corrected by about 0.8 per mil because of the fractionation that happens on there. So then I'm saying that that amino acid is minus 28.5 per mil. Okay, well, that's interesting. Well, it turns out that earlier last year, for some unknown reason, I analyzed the amino acid nutrients that are used to grow the Trichoderma. And they gave a isotope ratio of minus 21.4 per mil. It's very likely that these amino acids determine the isotope ratio of the amino acids in the amino acid pool in the fungus. And this difference here, then, is very significant. 7 per mil, which is exactly what Elsilla saw in his GC mass spec type of analysis. So this is reproducible. This difference is reproducible. No fluke of that experiment, and it's consistent with this analysis, in my view, of the differential isotopic content of AIB. Okay, conclusions from this part of the work. About 1 gram of the NMR pure alamethicin has been produced by a new method. Alanine and AIB are mostly separated by ion exchange chromatography, but there's a significant isotopic fractionation that occurs during ion exchange. And once you take that into account, the alamethicin AIB is 7 per mil lower than the isotopic ratio of the growth medium amino acids. Now, these results are consistent with, identical with the earlier isotopic analysis, and they're consistent with this theory of AIB biosynthesis that I'm promulgating. Well, okay, back to the stages of research. Okay, well, pretty much all 4 carbons, I think, have been analyzed, but the other ones are still a work in progress. Next year, I will, once I find out some more answers like this, I'll give another seminar where I've either proved or disproved my biosynthesis theory. And, okay, are there any questions now?